अस्सलाम वालेकुम सर।
अस्सलाम वालेकुम कोकब। अस्सलाम वालेकुम राफिल।
वालेकुम सलाम।
जी 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 कोकब, बस तकरीबन 5 मिनट में शुरू करते हैं क्योंकि थोड़े से लोग कम है अभी।
ओके सर। ओके सर।
हेलो क्लास स्टार्ट अभी तक क्यों नहीं हुई, किसी को पता है?
5 मिनट में।
अस्सलाम वालेकुम।
वालेकुम सलाम।
वालेकुम सलाम।
अस्सलाम वालेकुम। आवाज आ रही है?
जी सर।
Okay. Okay, so should we start?
Yes sir.
Okay. Today's our topic is a little bit different. What you expected is genetic engineering in agriculture. So actually, we are in a subject like molecular biology too, and that is more towards recombinant DNA technology. And recombinant DNA technology, where we will be dealing with different techniques and tools used in different fields regarding genetic engineering. And today we will be studying the genetic engineering that can be used and that are used in agriculture. Actually, this topic is too much extensive, too much practical, and too much explored. I don't know. I don't know how I could walk you through the lab experience and the lab techniques that are used, because this is too much practical actually. So I will be trying to add some videos regarding some of the techniques. Maybe you will be um, very well experienced with that. Maybe you have done that within the labs, but uh, this this is what I want to share with you. Okay, so to start with the Dolly sheep, this was the golden example of genetic engineering. And what was done there, a donor cell is taken from a sheep, and that was actually somatic cell. So somatic cell, that means the shortened chromosome. You already know what happening there in somatic cells, and what is the difference between there and the gametes and the somatic cells. So the chromosome will be shortened. So the donor cell was taken from there, and that was fused with a cell with a cell having no nucleus. So these two were fused, and they were then introduced in the uterus of a foster mother, and then the embryo produced, and that gave rise to a dolly sheep. So this was originally the mother was actually 10 years old, and it was said that the dolly sheep is actually 10 years old physiologically, and due to the chromosome length. So this was what happened there, and this revolutionized the field of genetic engineering. So after that, we will be talking about starting with bacterial transformation. So there will be some techniques like lying here, how we can engineer the genes, how we can do that. So first of all, we will be using bacterial vehicles like bacterial cell transformation, how bacterial cells could be transformed. So initially, the uptake, if this is the DNA we want to introduce in the bacterial system, what we'll be doing, the bacteria will take the DNA, and the bacteria that is capable of taking the DNA will be known as competent bacteria or competent cells. So competent cells will be those bacteria or those cells that is competent to take bacteria from the external environment. So the competent cells, so DNA may be integrated in the chromosomal DNA, or it can exist as a plasmid. So both can happen within the bacterial transformation. So two steps are important. First is making competent cells. So normally cells are not competent. They do not take bacteria, do not take external DNA. So we need to make them competent to take the bacteria and to take the genes from the outside. So and another is plant transformation. So that was bacterial transformation. And what is the plant transformation? We already have gene within the plasmid or within the DNA or linear DNA, and it will have uh, some gene as a marker. So both gene will be inserted into a bacterial cell. The whole plasmid will be taken into a bacteria known as Agrobacterium tumefaciens. So Agrobacterium has the capacity to deliver the gene of interest to the plants. So Agrobacterium, we will transfer this plasmid to Agrobacterium first, the bacteria. So the bacteria will take and will multiply and will replicate the plasmid, the DNA, and after it will also have its own chromosomal DNA. So this will integrate in there. This will have been in there as a plasmid. So the bacteria will have the gene, and it will transfer. We will grow that, that we will co-culture the bacterial cell with the plant cell. So once it is inserted in the plants, into the this is the plant cell, and it is the genome, and the bacterial this plasmid will be integrated into the genome of the plant, and those cells we will be multiplying on a media, and after that we will regenerate those plants, and we will get these transgenic plants. So transgenic means having this transgene. So we have transferred this gene so in the plant cell, and from that one cell we will be generating the whole plant, and that will be known as transgenic plant, or will be known as a GMO, means the genetically modified plant. So this complete plant is regenerated from transformed cells. So these are transformed cells, and we will be regenerating this whole plant. So basically there are two steps, two things: the bacterial cell transformation and plant transformation. So these two transformation will be focusing in this our this lecture. So any question up till now, or everything is clear?
Clear.
Clear है. So after that, Gigi.
सर हमने यहाँ पे हमारे पास जो प्लाज्मिड था प्लांट ट्रांसफॉर्मेशन के अंदर हमने प्लाज्मिड को रिकॉम्बिनेट किया, देन इसको हमने बैक्टीरिया में इंसर्ट किया। और सर ये जो बैक्टीरिया है ये हमारा पैथोजेनिक बैक्टीरिया है, ऐसी है?
जी जी जी
पैथोजेनिक बैक्टीरिया होगा
जी जी
सर। ये पैथोजेनिक बैक्टीरिया जो है इसने अपने जीन ऑफ इंसर्ट जो हमारा जीन ऑफ इंटरेस्ट था उसको इंसर्ट कर दिया उसने प्लांट सेल्स के अंदर, ऐसी है ना?
बिल्कुल
थर्ड
ठीक है,
पॉइंट के अंदर
बिल्कुल ठीक है।
तो सर ये जो हमारे पास है जो बैक्टीरिया था वो अब जो हमारा जीन ऑफ इंटरेस्ट जो होगा वो प्लांट के सेल में किस तरह इंट्रोड्यूस करेगा, ये बात थोड़ा कंफ्यूज कर रही है बस, बाकी सारा ठीक।
हाँ हाँ, ये आगे वैसे क्वेश्चन आपका अच्छा है इस स्टेज पे, ये आगे डिटेल से आ रहे हैं। इसमें असल में इस बैक्टीरिया में जो रीजन ये ट्रांसफर करेगा ना, इसको ट्रांसफर डीएनए कहते हैं। प्लाज्मिड का वो रीजन जो ये ट्रांसफर करेगा, दैट इज ट्रांसफर डीएनए, टीडीएनए। सो दैट इज, और इस पूरे प्लाज्मिड को ट्यूमर इंड्यूसिंग प्लाज्मिड कहते हैं, ठीक है। सो ट्यूमर इंड्यूसिंग प्लाज्मिड को पहले डिसआर्म्ड किया जाता है, मींस के उससे ट्यूमर इंडक्शन के जो प्रॉपर्टीज हैं, जो जीन्स हैं, वो रिमूव किए जाते हैं और जीन ऑफ इंटरेस्ट ऐड किया जाता है। सो बजाय इसके कि ये ट्यूमर इंड्यूस करें, तो ये अब जो वो जीन ऑफ इंटरेस्ट हमने ऐड किया हुआ है वो ट्रांसफर हो जाएगा। सो दिस इज द नेचुरल प्रोसेस। हमने नेचुरल प्रोसेस को मॉडिफाई किया है। हम वी विल बी टॉकिंग दिस अबाउट दिस लेटर ऑन इन दिस
सर सर
जी जी कोकब
सर हम कह रहे हैं कि ये जो प्लाज्मिड है ये बेसिकली ट्यूमर इंडोसिंग प्लाज्मिड है और हम इसे जीएमओस बना रहे हैं। बट सर जब हम इस प्लाज्मिड को इंट्रोड्यूस करवाएंगे तो ये एक स्पेसिफिक जगह पर जाकर हमारे पास इंसर्शन करवा सकते हैं, या अगर ये ट्यूमर है तो ट्यूमर भी एक स्पेसिफिक जगह पर ही करवाता है। तो सर इस तरह सारा का सारा प्लांट कैसे मॉडिफाई हो रहा है हमारा? अगर हमने सारा प्लांट मॉडिफाई करना है फिर तो हमें इसका सीड मॉडिफाई करना पड़ेगा ना।
हाँ, यू आर राइट, दिस इज टू बेसिक क्वेश्चन, बहुत अच्छा क्वेश्चन है। लाइक यहाँ पे हमने बात की ना कि हम एक सेल को ट्रांसफॉर्म करेंगे। जब एक सेल ट्रांसफॉर्म
sir.
होगा फिर उस एक सेल को हम मल्टीप्लाई करेंगे, बजाय इसके कि हम पूरा टिश्यू वो करें, ट्रांसफॉर्म करें। हमने टिश्यू को नहीं करना, सिर्फ एक सेल को, और बाकी
Yes, sir.
सेल जब इससे मल्टीप्लाई करेंगे वो इसके रेप्लिका होंगे। और जो प्लांट इससे बना है ना,
तो सर
इसका
हम
मतलब
इसे बेसिकली टिश्यू कल्चर करेंगे।
जी जी बिल्कुल रीजनरेट,
ठीक है।
इसको टिश्यू कल्चर ही करेंगे। मींस सेल कल्चर है, सेल्स और सेल से फिर पूरा प्लांट टोटिपोटेंसी के थ्रू पूरा प्लांट बनाएंगे, रीजनरेट कराएंगे।
ठीक है सर।
शुड वी गो फर्दर?
यस सर।
ओके, तो आज का टॉपिक बहुत इंपॉर्टेंट है, क्योंकि इसमें हम कुछ टेक्निक्स कवर कर रहे हैं। ठीक है, सो be attentive and try to focus these things, and it's too much important. आप लोग ने इस स्टेज पे क्वेश्चन कर लिया, that is too much to the point, relevant to the point ना। ओके, अब नेक्स्ट जो है ना, बैक्टीरियल ट्रांसफॉर्मेशन इज अ प्रोसेस ऑफ हॉरिजॉन्टल जीन ट्रांसफर। सो वी आर टॉकिंग अबाउट बैक्टीरियल सेल ट्रांसफॉर्मेशन। सो जिस चीज को हम ट्रांसफॉर्म कर रहे हैं ना, उसको ट्राई करें कि उसको पहले लिखें। ठीक है, so this will mean what we want to say. So bacterial cell transformation hai, so is a process of horizontal gene transfer by which bacteria take up foreign genetic material and from the environment. So it is not vertical gene transfer, it is horizontal gene transfer. So it is actually horizontal gene transfer, and not all bacteria are competent. So that means that every bacteria cannot take up the DNA. So that means competent cells preparation is important. So not all bacteria are competent, means capable of taking up exogenous DNA from their environment. The practical approach to acquire competent cells is to make bacterial cells artificially competent. So what will we be doing in the labs? We will be making these cells competent. हम bacterial cell लेंगे, हमने उनको खुद competent बनाना है using two types of methods. Either we will be going to use chemicals or electrical pulses. So these two methods we use to make the cells competent. हमने foreign DNA introduce करना है, तो हमने पहले उस सेल को कॉम्पिटेंट बनाना है। तो कॉम्पिटेंट बनाने के लिए, इस सेल को कॉम्पिटेंट बनाने के लिए हमारे पास टू मेथड्स हैं। द फर्स्ट वन इज थ्रू केमिकल्स, इसको हीट शॉक मेथड भी कहते हैं, एंड सेकंड वन इज इलेक्ट्रिकल पल्स। सो दिस इज मींस इलेक्ट्रोपोरेशन के थ्रू हम ये करेंगे। सो chemical induction of competent cells involves competence involves the following steps. First, सबसे पहले the cells should be chilled in the presence of calcium phosphate to make them permeable. तो calcium phosphate जो है ना, वो इसके cell wall के साथ bond बनाएंगे, and the chilling effect, and these two will be making these the walls of the bacteria permeable. So और उसके बाद हमने क्या करना, इंक्यूबेट, सबसे पहले चिलिंग टेम्परेचर पे रखना है विद कैल्शियम फॉस्फेट, और उसके बाद कुछ लोग मैग्नीशियम क्लोराइड भी यूज करते हैं। तो और उसके बाद इंक्यूबेशन विद डीएनए के साथ हमने उसको इंक्यूबेट करना है, मींस इन दोनों को अब मिक्स करना है। So mixing the competent, so these are the competent cells, so mixing the competent cells with the DNA, and then we will be giving heat shock, means उसको 42 डिग्री सेंटीग्रेड पे यकदम इन सेल्स को जो डीएनए के साथ हमने मिक्स किए हुए हैं, यकदम 42 डिग्री सेंटीग्रेड पे 60 to 120 मिनट सेकंड्स के लिए रखें, मींस 1 मिनट, लो बाजुकात people will be doing for 2 minutes. So उसको 42 डिग्री सेंटीग्रेड पे रखें और यकदम दोबारा फिर चिल कर दें। So this will be heat shock, means the cells will be opened due to the temperature, and once the cells will be opened, the DNA will go enter into the cells, and उसके बाद हमने दोबारा इसको chilling temperature will be back onto chilling temperature. So that causes the DNA to enter the cell. So this will be transformation. So competent cells बनाए, उसके बाद हमने heat shock method दिया, heat shock दी, and that makes the DNA enter the cells. So this is the chemical induction or chemical transformation method है, bacterial transformation. So अगर इस method को हमने use नहीं करना, we can use another method. The bacterial cells are made permeable by subjecting them to electrical pulse. a process known as electroporation. So हम इसको electroporation के through भी ही use करेंगे. इस केस में हम कैल्शियम फॉस्फेट या केमिकल्स ये यूज नहीं करेंगे. इस केस में electroporation के थ्रू हम ट्रांसफर करेंगे, बैक्टीरियल डीएनए को ट्रांसफॉर्म करेंगे. जी जी. Any question? Any question?
सर ये जो आपने लास्ट में बोला इसकी समझ नहीं आई, आपकी आवाज कट गई थी.
ओके, सो मैं ये कह रहा था कि कौन सा दिस लास्ट पॉइंट, दिस पॉइंट?
यस सर, इलेक्ट्रोपोरेशन वाला.
ओके, सो यहाँ पे एक्चुअली हमारे पास 2 मेथड्स हैं. फर्स्ट वन इज केमिकल. सो केमिकल के थ्रू हम सेल्स को ट्रांसफॉर्म करेंगे. लाइक केमिकल्स में हम क्या करेंगे, सेल्स को चिलिंग टेम्परेचर पे रखेंगे, कैल्शियम फॉस्फेट के साथ ट्रीट करेंगे. दिस विल मेक द सेल कॉम्पिटेंट. इट विल बाइंड टू द सेल एंड दैट इट विल मेक देम पोरस. इसके बाद मीनवाइल हम उसको डीएनए के साथ मिक्स करेंगे. जिस डीएनए को हमने ट्रांसफॉर्म करना है उसके साथ सेल्स को मिक्स करें और उसके बाद क्या है, उसको हीट शॉक दें. हीट शॉक देंगे 42 डिग्री सेंटीग्रेड पे 1 to 2 मिनट्स के लिए. तो उसके बाद क्या होगा, दी डीएनए विल इंटर और उसके बाद हमने सेल्स को एकदम दोबारा चिलिंग टेम्परेचर पे रखना है. सो दिस हीट शॉक विल मेक द डीएनए इंटर इनटू द सेल एंड द पोर्स विल क्लोज अगेन. सो अब दिस इज द केमिकल इंडक्शन मेथड. Another, अगर हमने chemical induction method use नहीं करना, we can use electroporation method. So electroporation is also known as using the electrical pulses or electrical pulse को हम use करते हैं. और इसमें क्या होता है कि the cells can be permeable through the electrical pulses, and in this case we will not be using calcium phosphate or magnesium chloride or these salts. हम ये salts use नहीं करेंगे electroporation में. So up till now it's clear?
Yes sir.
Okay, so okay, we will be having a video regarding chemical methods, the bacterial transformation through chemical methods, and if I can share the sound. And let's see how this happens within a lab. This video is not much. I couldn't find some professional video regarding it, but this is what we do in the lab. Okay, so this is a good video regarding chemical. So this is bacterial transformation regarding chemical methods. So bacterial transformation through chemicals. How we do this? This is one of the protocol, and this is the competent cell transformation. So let's check this.
This video will walk you through the basics of chemical.
Can you hear the voice of the?
Yes, sir.
Okay. So
Transformation. Transformation is the process by which bacterial cells take up foreign DNA from their environment. Typically, this is done in the lab for 2 main reasons: to propagate a recombinant plasmid or to obtain the results of a subcloning reaction. There are 2 main classes of competent cells: chemical and electrocompetent. The procedures shown in this video can be used with most chemically competent cells. We are focusing on the protocol provided with Invitrogen's top 10 strain of comp cell. First, let's talk about storage conditions and what comes with your kit. Competent cells are stored at -80 degrees. Invitrogen's kits come with vials of competent cells, transformation instructions, a vial of SOC medium, and a PUC-19 transformation control. The one-shot top 10 kit used in this video comes with vials containing 50 microliters of competent cells, enough for 1 transformation per tube. The competent cells must remain frozen until just before you are ready to use them. If they thaw too soon, it can affect the transformation efficiency. For your transformation protocol, you will need the following items: a water bath set at 42 degrees, an ice bucket with ice, a 37-degree shaking incubator and a 37-degree incubator, 10 centimeters diameter LB agar plates with the appropriate antibiotic. In this case, a 100 micrograms per milliliter of ampicillin. The selective plates should be warmed in an incubator for 30 minutes before you use them. You will need 1 plate for each transformation. Finally, you will need the vial of SOC medium thawed and at room temperature. The first step is to mix the comp cells and the plasmid DNA of interest. Briefly centrifuge the DNA and put it on ice. This DNA can be from a ligation reaction or plasmid DNA you wish to propagate. Next, thaw on ice 1 50 microliter one-shot vial of cells for each transformation reaction. Pipette 1 to 5 microliters of your DNA samples directly into each vial of competent cells. Mix by tapping gently and not by pipetting up and down. Your remaining ligation reaction can be stored at -20 degrees. We also highly recommend setting up a transformation control with the PUC19 provided to ensure the transformation was performed correctly and the competent cells were at the expected competency. Add 1 microliter of the PUC19 control plasmid to 50 microliters of competent cells and tap gently to mix. The next step is to incubate the cells on ice for 30 minutes. Next is the heat shock step. This is harsh on the cells, so be sure not to mix or shake the vial. Incubate for exactly 30 seconds in the 42-degree water bath. After the incubation, remove and place on ice. Now the cells are allowed to recover from the transformation and are grown in rich SOC medium. Add 250 microliters of the pre-warmed SOC medium to each vial. Next, place the vials in a microcentrifuge rack inside the shaking incubator, or tape the vials on their sides to the shaking platform in the incubator. Shake the vials at 37 degrees Celsius for 1 hour at 225 RPM. If you do not have a shaking incubator, you can add the vials to a 37 degrees Celsius regular incubator. Once the cells have recovered, it is time to plate them. The antibiotic you added will select only transformed cells. Pipette 15 microliters from the individual vial onto its own labeled LB plate. The remaining transformation mix may be stored at 4 degrees and plated the next day if desired. For the control, remove 10 microliters from the vial and mix with 20 microliters of fresh SOC and plate the entire 30 microliters as you did with the other transformations. To spread the cells evenly across the plate, add 5 to 8 sterile glass beads and rotate, or use a sterile glass rod to spread across the plate. After removing the glass beads, invert the plates and incubate in 37 degrees overnight. Plasmid containing the antibiotic resistance marker that allowed them to grow in the presence of the selection antibiotic. The PUC19 control plate will have a larger number of colonies than your ligation plates. Many people also use blue-white screening for selecting their clones. Each of these is a transformant and has taken up the plasmid containing the antibiotic resistance marker that allowed them to grow in the presence of the selection antibiotic. The PUC19 control plate will have a larger number of colonies than your ligation plates. Many people also use blue-white screening for selecting their clones. So what happens if you go to the incubator and your transformation did not work and you have no colonies? Transformation works almost every time, but occasionally it doesn't. Here are some areas to check. ensure the antibiotic in the agar plates matches the clone resistance marker and is at the correct concentration. Impurities in your DNA sample can also affect transformation. Ensure phenol, proteins, ethanol, and detergents have been removed from the DNA. Ensure there is not an excess of DNA or volume. Also, ensure the competent cells have been handled correctly. Use immediately after thaw and do not vortex. Ensure the incubators and water baths are at the correct temperature setting. For more troubleshooting tips or information on all competent cell strains, visit www.invitrogen.com/compcells.
So is it clear? Is it clear?
Yes, sir.
Okay,
Yes,
any
sir.
question regarding it?
Yes, sir.
So this is how a chemical method works. So chemical method, लेकिन इसमें एक चीज है कि इन्होंने तैयार competent cells लिए थे. इससे पहले competent cells को बनाया जाता है, so they can be made with treating the cells with calcium chloride or the calcium phosphate or the magnesium chloride. So after that, we will be going for the chemical method of transformation. So this is transformation. So and what about the electroporation method? So this video is not professional, but I didn't found any video that could walk us through the lab. So this is an optimal, but this is what we are actually doing in the lab for electroporation method. So this is the electroporation. So let's check this out.
We're going to be doing, here, let me move you this way so Josh can get in there. We're going to be doing an electroporation, and we're going to be electroporating some linear DNA so that we can do a Dapsonko-Weiner and do a genome integration. So I've just done my final spin down of my competent cells with glycerol. I'm just going to pull off any of the excess stuff, hoping to get rid of as many contaminants as you want. And I've learned the hard way recently that that's a very important step. Don't neglect that step, or you will lose a lot of efficiency. You're not going to get nearly as many clones with a dirty competent cell prep than you will with a clean competent cell prep. So I've rinsed these off many times today. I think this is like the fourth rinse step that I've done, and I still have a pretty good cell pellet. So I'm just going to pull off any of that excess stuff. I'm going to pull off some of the cells, but as long as I still have the majority of it there, I'm going to be good to go. So now what I have is DNA that I want to integrate, and I have cells. And I need to get that DNA into the genomes of those cells. That will be accomplished using a cuvette. What I'm going to do is I'm going to put those cells into this here cuvette. I'm going to put my DNA, I'm going to mix my DNA with those cells. I'm going to zap them with electricity, and through magic, the DNA is going to go inside of those cells. And those cells already have a plasmid called PKD46 that has a bunch of proteins that will essentially, just for our purposes, we're just going to say it'll integrate whatever linear DNA is in there. Normally, you can get away with a smaller prep, about 1 mil, but since I have a big piece that I'm trying to put in there, it's about 6 kb, 6,000 base pairs, I need to do a slightly larger scale prep. So I'm doing a mid-scale prep, 50 mils. So, let's see here. I am, kind of lost track of what I'm doing. Oh yeah. So we have a cell pellet here, and I need to resuspend this. Each Dapsonko-Weiner, or rather each electroporation, takes 100 microliters of competent cells and a few microliters of DNA. So since I have 3 samples that worked over here, we just ran a gel and we found out that these 3 samples actually have DNA in them. I'm going to want to be running about 3 electroporations per sample, and I have 2 different samples. So 2 strains, 3 samples, 6 total. I have 4 in here. I'll go get some in a little bit. So I have 100 microliters. I have my tube. This is our MZ1061 cells, and I'm going to go back to my 10% glycerol. I'm going to put in 3 volumes of 10% glycerol. So each volume is 1 transformation. And then I'm going to close it up. And I'm going to do the same thing for our other strain, which is our 9116 cells, which are just a strain that we have in the lab that I use a lot. So again, 1, 2, 3. And I'm actually going to do a fourth one for good luck. Sometimes you get very unlucky and you get a spark. We call that an arc. That's because you had too many salts and you basically electrocuted your cells. You put them on the electric chair and they're all dead and you don't get anything. So I'm going to resuspend those real quick. Those are all nice and suspended. That's kind of what it looks like, a little bit milky. And it goes right back on the ice. Now in order to do the actual transformation, you're going to need a couple things. I appear to be out of 1.5 mil tubes, so I'm going to steal some from my good neighbor here. You will never know. And one of the tubes goes on ice. One of the tubes goes on the side. I'm going to get my P1000. I'm going to set up my recovery. So I'm going to set this to 250. I'm going to put this on there. Set it aside for now. I'm going to be using all of my pipettes. And this pipette is already set to 100. So that goes there. And set it aside. And then I'm going to grab, finally, my P20. And this is how much DNA I'm going to use. I'm going to go with 4 microliters. Oh, we'll try 4 microliters of DNA. See how that goes. So take a tip, get 4 microliters of DNA. This is just linearized, cleaned up PCR product. I'm sorry, this is just cleaned up PCR product, which is a linear DNA. I put my 4 microliters at the bottom of that tube, which has now been chilled. Goes back on ice. I grab my pipette, my P200, and I'm going to pull up 100 microliters of cells. Take a look at it, make sure there's no bubbles. And then I mix these two in this, so I just go up and down. And again, this whole process, you want to make sure it stays cold and you don't want to have any bubbles. So then I bring it all back up. That gets tossed. I grab a cuvette. And then there's a little notch in there. I'm going to put my pipette in that notch and slowly eject. And I don't eject all the way because that will make bubbles. So I get rid of my tip, tap this down. This goes back on ice. And now we're almost ready to go. The last thing I need to do is get my recovery. So I'm going to take some plain 2IT. This has no antibiotics in it. I'm going to hold it up to the light, check to see that there's no contamination. These are very susceptible to contamination because there are no antibiotics. And I should be doing this with a flame. I'll just set that up right now. Again, you always want to keep a flame next to stuff that doesn't have any antibiotics. Pull up our 250 microliters of 2IT. This is a very rich medium. And then we're going to grab our tube. I need a Kim White. So we're making... Grab a Kim White. Grab your cuvette. And then we're going to run on over to the electroporator. Cuvette goes on top. Tube goes on top. Kim White gets placed there. Put our recovery up there. We're going to check volts 1600. Then we're going to go back to time constant. I'm doing this slowly. I should be doing this a lot faster because otherwise it'll warm up. I'm going to really thoroughly dry off my tube. Tap it down. And then put it in this holder. There's a little knob on the side. That knob goes down to that little notch. Throw that in there. Press pulse twice. It'll make a noise. And if we don't hear a pop, then we know it worked. 5.8 is good. We generally want to get above a 5.0. I'm immediately going to stick my tube or my 2IT in there, resuspend it, and this is going to go for recovery. So I'm then going to start to pull it out. So it just goes in that tube that we got over. And you want to try to get as much of it as you can. And then this should immediately go at 37 degrees and give it a chance to recover. So I'm going to label this as what my sample is and what my time coefficient was. So grab a pen. Where are my pens? So So this is sample number 2, time coefficient of 5.8. And then this goes in the incubator. And then we just do the same thing with the rest of our sample. So I'll just do that one more time, and I'll give a much briefer explanation. So we're going to do 4 microliters of sample number 3. I'm going to grab my 2 tubes. So DNA goes into tube number 1. Check that tip. Grab my, where did my deciliters go? Oh, my pipettes are all over the place.
After that, he will repeat the whole procedure again, and that's it. So I think you are well versed with the electroporation method as well. Is it clear?
Yes, sir.
Yes, sir.
Okay. So we are done with the 2 methods. The first one is a chemical method, and the second one is electroporation. So the chemical method and the electroporation. So now before this we were talking about bacterial cell transformation. So why plants? So bacterial cell transformation, you already know that is for different purposes, to make the recombinant DNA multiply in a cell. So now why a genetic engineer a plant? So why plant genetic engineering? So better yield and quality of crops, so to increase the quantity as well as the quality. So we gonna go for genetic engineering, produce super crops. So we're gonna produce super crops. We want to produce super crops for to nourish our increasing population. So we are gonna produce this through genetic engineering. And the next is a microorganism can be used as a pollutants cleaner. So we can use the microorganisms for pollution cleaner. So we can also use plants as a pollution cleaner. So bioremediation of polluted environment. So we can use bioremediation. So we can also use phytoremediation. So the plants could be used for remediating the environment. So polluted environment. So we can use them as well. So these are this is why we use plant genetic engineering. So why create transgenic plants? And the first thing is to improve agriculture value of plants. So increase yield, they increase yields through herbicide resistance, through pest resistance, enhance nutrition quality. Nutrition enhancements is also one of the um, goal to improve or to create transgenic plants. Enhance taste of a crop or of food. So these are the some of the objectives for which we use transgenic plants and for which we create transgenic plants. So this is the golden example. Actually, the plant can produce proteins for human needs. So pharmaceutics, so we can use them. So golden rice produced through genetic engineering. So golden rice was produced. Was through genetic engineering. The color of this rice is golden now. Rather than white. So this golden rice is actually, and this is a gene was transferred, and that gene was beta carotene, and beta carotene is actually a precursor for vitamin A, and due to its color the grains become golden in color, yellowish golden. So this become golden in color. So that was golden rice. So golden rice is one of the example of this transgenic plants and within the field. So we are within the market, we have this golden rice, this transgenic golden rice. So edible vaccine is another application of these transgenic plants. For instance, we are taking oral vaccines, and for instance, that is some protein, and we can transfer that protein and their gene into a plant, and now we are taking fruits like banana or apple, and that banana or apple have the vaccine within it. So by taking a banana or an apple, we are taking edible vaccine, or we got vaccinated. So this can happen, and this is how people are doing with these transgenic plants. So after that, modified plants can be used to study effects of genes. So to study the effects of genes, so we also can use plant to transform them, and we make them transgenic. So after that, what is actually the basic concept and the basic difference from other organisms? So an entire plant can be regenerated from a single cell, and this is the Totipotency. So a single cell can give rise to a whole plant. So this is Totipotency, and this Totipotency, the efficiency of this thing of the plants has been taken off and was utilized for generating transgenic plants. So no separation of germs and somatic cells are there in plants. So in animals, this can happen that the germ and somatic cells can be differentiated, but in plants there is no such case. So the germ and somatic cells cannot be differentiated as well, because we said every cell can give rise to the whole plant. So with this concept, actually, the germ cells can give rise, in our case, in humans and in animals, the germ cells can give rise to the whole organism, and not the somatic cells, but in plant, the somatic cells can give rise to the whole organism naturally. So production of crops for better yield and quality. जी
सर यहाँ पे ये जरा साइड पे जाइएगा जरा प्रीवियस स्लाइड
जी
सर। यहाँ पे जो गोल्डन राइस है सर हमारे पास, हमने एक बार जरा का प्रोसीजर समझा देंगे सर। देखो जेनेटिक इंजीनियरिंग सर, हमने यहाँ पे जो यूज कर रहे हैं बीटा कैरोटीन, ये विटामिन का प्रीकर्सर है। तो फिर यानी कि कैसे आगे
ओके
ये विटामिन क्या?
ओके इस तरह किया गया था इसमें कि ये राइस लिया गया, प्लांट राइस का, और विटामिन ए का प्रिकर्सर जो है बीटा कैरोटीन है, ठीक है। बीटा कैरोटीन का जीन जो है ये ट्रांसफॉर्म किया गया विद इन राइस में। अब राइस जब प्रोड्यूस होंगे तो उसमें बीटा कैरोटीन प्रोड्यूस होंगे। बीटा कैरोटीन इज अ प्रिकर्सर। जब इसको हम यूटिलाइज करेंगे तो इसने कन्वर्ट होना है विटामिन ए में, ठीक है। सो ये राइस उन इलाकों के लिए बहुत अच्छा है वेयर देयर इज डेफिशिएंसी ऑफ विटामिन ए। सो ये अफ्रीका में प्रोवाइड किए गए, इस तरह ये बहुत ज्यादा पॉपुलर है गोल्डन राइस to cater the deficiency of vitamin A. So rather than taking tablets or multivitamins like for vitamin A, we are taking golden rice, and that is catering the deficiency of food as well as for the vitamin A. So similar is the case with other things. इस तरह आयरन को भी किया गया, आयरन के प्रिकर्सर्स को लिया गया, आयरन को जो पाथवे, फॉलिक एसिड के पाथवे में जो चीजें यूज हो रही हैं उनके जीन्स को ट्रांसफॉर्म किया गया है, एंड पीपल आर यूजिंग दैट। जी
सर ये जो भी हम लोगों ने बीटा कैरोटीन की जीन ट्रांसफर की है, तो सर इससे जो नेचुरल मतलब प्रॉपर्टीज हैं प्लांट की, उस पर क्या चेंज आ सकता है? एक तो हमें कलर का और चेंज आ रहा है उसकी वजह से, लेकिन उससे वो कोई इफेक्ट नहीं है हेल्थ पे या हमारी
ये कलर असल में बीटा कैरोटीन की वजह से है।
यस सर। बट सर ये जब हमने जीन ट्रांसफर किया है तो हमने डेफिनेटली राइस के जीनोम में ट्रांसफर किया, तो जीनोम में कोई ना जीनोम में डेफिनेटली डिफरेंस आया, तो जीनोम का डिफरेंस जो है स्पीशी पे क्या है सिवाय ये कलर और प्रिकर्सर?
बिल्कुल ये चीजें हम आखिर में रिस्क में करेंगे डिस्कस। इस वजह से बहुत से लोग जीएमओस के अगेंस्ट भी हैं, हमने वो चीजें डिस्कस करनी है वहां पे। ये असल में यहाँ पे जहाँ पे आपका क्वेश्चन है ना, दिस इज हिट एंड ट्राई। आपके पास मींस हमने पहले भी पढ़ लिया है ना कि एक सेल को ट्रांसफॉर्म करें, लेकिन फिजिकली एक सेल ट्रांसफॉर्म नहीं होता, बहुत सारे सेल ट्रांसफॉर्म हो जाते हैं। फिर उन सेल्स में सिलेक्शन की जाती है, मींस 10-15 सेल्स हमारे पास आ गए, उससे 10-15 डिफरेंट प्लांट्स आ गए। नाउ एवरी प्लांट इज अ सेपरेट लाइन। अब सारे प्लांट जो है वो हम उनको सेपरेट कंसिडर करेंगे, उनसे जब सीड आएंगे फिर उनको अलग-अलग लेंगे। अब ये देखना है कि ये जीन ऑफ इंटरेस्ट जो हमारा बीटा कैरोटीन है ये कितनी दफा उसमें है, मींस के जीनोम हो सकता है सेल के जीनोम के अंदर 3-4 दफा वो इंटीग्रेट कर गया, मीन 3-4 कॉपीज हो, कि बाज लाइंस में एक कॉपी होगी, बाज लाइन में ज्यादा होंगी, बाज लाइंस में वो उसमें फिनोटिपिकल इफेक्ट्स आएंगे, क्योंकि उसके हो सकता है कोई इंपॉर्टेंट जीन्स जो हैं वो डिस्टर्ब हो रहे हैं उसकी वजह से, इंटीग्रेशन की वजह से। So there are many possibilities that will be dealing with some practical exposure of those plants ना। ठीक है, आवाज आ रही मेरी?
ओके सर। यस सर।
Okay,
Yes,
so
sir.
should we go further?
Yes, sir.
Okay, so राफे आज खामोश है राफे.
सर
Okay, so अब देखें
सब क्लियर है.
clear है, that's good. So production of crops and bacterial quality, quality and quantity. So इसमें क्या है कि genetic engineering has helped to increase crop productivity by introducing qualities such as disease resistance, increased drought tolerance. So इसमें बहुत ज़्यादा काम हुआ है and people are coping with the disease resistance in crops and the drought tolerance, because to make plants for those areas where there is drought है, um, the plants cannot be grown there, वहाँ पे plant grow नहीं हो सकते. So वहाँ पे फिर क्या किया जा सकता है? So इस तरह के इलाके भी हैं जहाँ पे plant grow नहीं हो सकते, जहाँ पे पानी नहीं है, ठीक है. So वहाँ पे हम देखते होते हैं कि like cactus और इस तरह के plants वहाँ पे grow किए जाते हैं. फिर वहाँ से उन cactus और उनसे जो drought tolerance के genes हैं वो लिए जाते हैं. They transform those genes into our crops में transform किए जाते हैं. Now crops can be grown there. So this is the major field of research कि वहाँ के लिए even disease resistance के लिए भी genes को transform किया जाता है within the plants to get better plant resistance and to increase the drought tolerance, salt tolerance के लिए भी काम किया जाता है. And there are many fields where people are working through genetically modified organism GMOs, and they are producing plants. So genes from naturally drought resistant plants are used to increase drought tolerance in plants. So this is the variety you can see. So this is Zenobia seeds in collaboration with Sari seed developed by AGRA. So this is an African variety, and this has been grown. So this will be just like normal, बिल्कुल normal की तरह होंगे, and there will be no difference phenotypically. आपको कोई difference नजर नहीं आएगा, सिर्फ एक gene transform किया गया है and that's it. So two methods are there. या तो ये किया जाता है ना, like कोई resistance variety हो, उसमें nature में कोई resistant variety, for instance this is maize ना, maize के जो resistant varieties हैं and that can be wild type varieties जो resistant हो किसी disease के लिए, उनको cross किया जाता है basically. जो कन्वेंशनल मेथड है क्रॉस किया जाता है, उसके जो ऑफस्प्रिंग आते हैं फिर उनको देखा जाता है कि उसमें अब यील्ड भी ज्यादा हो, क्योंकि यील्ड तो 50% रिड्यूस हो जाएगी. तो दैट हैपेंस, और उसमें सारे जीन्स 50% एक जीनोटाइप से आ जाते हैं, फिर 50% वाइल्ड टाइप से आ जाते हैं. सो उससे बेटर है कि 50% ना की जाए, जस्ट वन जीन ट्रांसफॉर्म किया जाए. एंड दिस इज द सुपीरियरिटी ऑफ दिस GMOs प्रोडक्शन ओवर द कन्वेंशनल मेथड्स. So, and this is easy to go with it. So after that, this is within the field, और ये market में है अगर हम इसको देखें. So this is within the market, and we are having genetically engineered papaya. Sanap के नाम से है एक. So this is resistant to papaya ring spot virus. So this papaya ring spot virus से ये अब resistant है. And home grown papaya that is affected by papaya ring spot virus. Papaya ring spot virus के इस तरह के spots आते हैं, ring spots. ठीक है. So now this is resistant to that virus. Papaya में virus का बहुत ज़्यादा issue है, and that reduce the grown, the yield as well as the taste of the and taste of the fruit. So ये sanap जो है, this is genetically engineered. So अब देखें, it has no virus. So in the market there are many options, and people are working on that continuously. So after that, so another example, the golden example is BT cotton. You have heard about that because this is native to our region as well. So cotton is here, grown here as well. So Bacillus thuringiensis, this is B and T, Bacillus thuringiensis, BT. So this is a bacteria, Bacillus and thuringiensis, this is the genus and this is the species. So Bacillus thuringiensis is a bacteria, and this bacteria actually produces insecticidal crystal protein. So ICP, this produces that, and also known as cry protein. And this cry protein is actually endotoxin. So what the basis is, there is a bacteria known as Bacillus thuringiensis, and this Bacillus thuringiensis has an insecticidal crystal protein known as ICP, also known as cryoprotein, also is an endotoxin. So this is the basis. So we have this thing in the nature, and this gene was taken from the bacteria and was introduced into cotton. So this endotoxin, when introduced into the cotton, what happens then? So there is a major problem in cotton, that is the bollworm. This is the adult of that bollworm, and this is the larvae of that. So in larval stage, this is more destructive because this feed on the cotton, and this is more destructive. So what happens? So this is normal cotton. So normal cotton is okay, and when this is bt cotton, so the bt gene is transformed into cotton, and this is bt corn. So bt gene has been transformed into cotton, and what it will be having, it will be having this bt toxin, this endotoxin. So this can be from the bacteria, this can be now from the plant. So because the plant is expressing the same gene. So when this is ingested by the larvae, it what will be happening there? There it will be, and the insect midgut cells will take up that, and this will give rise to septicemia, and that is the blood infection. So the toxicity in the blood. So this larvae will die, and the cell death will occur there. So this is how this natural thing has been introduced into plants, and the plants now, these plants are now resistant, and no larvae can feed on these plants now. So after that, this was the example from the first example from what was from papaya. This was from cotton. So after that, so technology of production, how to produce these transgenic plants. So main steps for developing transgenic plants. So there are several main steps. There are several steps, and the first one is identification of effective gene. So we need to identify the gene, where the gene is present, what the gene is, and what do we want. So our objective. So we need to have our objective. So the effective gene we need to have is to identify first our problem, and then we need to amplify that gene and to have that gene. So now the gene transfer. That second step is the gene transfer, and the gene transfer into the plant. So for to transfer the gene into the plant, we need to have it in the agrobacterium. So for agrobacterium, we need to have the recombinant plasmid. So for recombinant plasmid, so we need to clone that into a plasmid to transform bacteria, agrobacterium, and after that we need to transfer those into the cells of plant cells. After transformation, we need to regenerate from the cells, and that could be from protoplast, the cells without having the cell wall is protoplast, or the callus. So these are the unidentified, undifferentiated mass of cells. So these are undifferentiated mass of cells. So this will be the mass of cells that we cannot differentiate. Either the cells are of roots, or stem, or of other organs. So that will be undifferentiated mass of cells. Or we can also regenerate from tissues. So we can take a leaf tissue and we can regenerate that. We can regenerate whole plant from leaf tissue, from stem tissue, from any tissue of a plant. So first one is identification, second one is the gene transfer, third is the regeneration, and the fourth one is expression level of the desired gene at a desired level. So we need to identify either the gene we have introduced into a plant, either that is expressing or not, either that is producing, if we need proteins of that. So either protein is produced or not. If we need only RNA, either RNA is produced or not. So we need, and the desired level. Maybe we need more copies. Maybe we need more number of that specific protein or specific gene, but that is not producing at that same level. So we will not be taking that. So gene expression is at a desired level is too much important. So, and fifth one is field test. either it is expressing in the field, either it is not producing any problem in the field. So we will be having field test, and after that we will need to have approved that for commercialization process. So we will also need to have it approved. So these are the 6 steps that involved in developing any transgenic plants. So any question regarding it?
सर ये फील्ड टेस्ट क्या होता है?
फील्ड टेस्ट ये है ना कि हमारे पास तो लैब में हमने एक्सपेरिमेंट कर लिया, जीन एक्सप्रेशन हो रही है, सारा कुछ ठीक है, हमने उसको अब फील्ड में भी टेस्ट करना है, बाहर ओपन एनवायरनमेंट में, ठीक है। ओपन एनवायरनमेंट में आइदर ये तो नहीं है कि वो प्लांट जो है ग्रो ही नहीं हो सकते, लाइक लैब में कंट्रोल एनवायरनमेंट में ग्रो हो सकते थे, लेकिन जब हम उसको फील्ड में लेकर आए हैं ओपन एनवायरनमेंट में, क्योंकि वहाँ पे स्ट्रेसेस भी होंगे डिफरेंट टाइप्स ऑफ, सो वहाँ पे वो ग्रो ही ना कर सके, वहाँ पे वो डिजायर्ड लेवल पे जीन एक्सप्रेस ही ना वहाँ पे वो ग्रो ना हो सके, कुछ मल्टीपल फैक्टर्स हो सकते हैं। हो सकता है वो प्लांट अब बहुत ज्यादा ससेप्टेबल हो मल्टीपल डिजीजेस के लिए, सो उस प्लांट पर डिजीज अटैक बहुत ज्यादा हो, एक जीन के हो सकता है कुछ जीन्स उसमें इंटरप्ट हो गए हो, कुछ जीन इंटरप्ट हो गए हो, सो वी नीड टू हैव देम टेस्टेड इन द फील्ड, एंड आफ्टर दैट वी नीड टू अप्लाई फॉर कमर्शियलाइजेशन। इसमें भी बहुत सारे स्टेप्स हैं वैसे।
ओके सर,
ठीक है, सो शुड वी प्रोसीड फर्दर?
यस सर।
यस सर।
Okay, so how to get a gene into a plant? So there is a plant transformation. So that could be, so different methods हैं जिसके थ्रू हम plant transformation कर सकते हैं। Like इससे पहले हमने देखा था ना कि bacterial transformation हम 2 methods से करते होते हैं। Like the first one is through heat shock method or chemical method, and the second one is electroporation. और यहाँ पे जो प्लांट ट्रांसफॉर्मेशन है हम इसको भी 2 मेथड से करते होते हैं, इवन 3 मेथड, इनफैक्ट भी एक मेथड है पॉलीथेरिन ग्लाइकोल का, लेकिन यहाँ पे हम 2 को पढ़ रहे हैं। द फर्स्ट वन इज थ्रू एग्रोबैक्टीरियम। सो एग्रोबैक्टीरियम नेचुरली क्या करता है, सो एग्रोबैक्टीरियम अगर प्लांट में कहीं पे वूंड है, so the agrobacterium will be induced and will infect this plant and will induce a crown gall there. So crown gall tumor produce करता होता है वहाँ पे, and this is natural, और agrobacterium soil borne bacteria है, so ये soil में रहता है, and once it is the wound is found, so this will attack there and it will induce the cells to grow. So आफ्टर दैट दूसरा मेथड जो है वो माइक्रो प्रोजेक्टाइल बम्बार्डमेंट है। दिस इज माइक्रो प्रोजेक्टाइल, दिस इज द इक्विपमेंट। इसमें हम माइक्रो प्रोजेक्टाइल बम्बार्डमेंट के थ्रू क्या करते हैं कि हमारे पास प्लाज्मिड्स हैं, प्लाज्मिड के साथ हम गोल्ड पार्टिकल या टंगस्टन पार्टिकल्स के साथ हम मिक्स करते हैं, और उसके थ्रू हम जीन्स को, हमने अगर हमारे पास कैलस है या टिश्यू है या कोई भी एक्सप्लांट है, हमने उसको बम्बार्ड करना है इनटू द सेल्स, ताकि ये जो डीएनए है वी विल वांट इट टू रीच टू द न्यूक्लियस, ये न्यूक्लियस तक जाए, और वहाँ पे क्या हो, फिर उसके बाद हमने उस प्लांट को रीजनरेट करना है, उससे होल प्लांट रीजनरेट करना है। So this is how things occur in microprojectile. So now in this case we don't need
agrobacterial. Microprojectile दोबारा बता दो।
So microprojectile, अच्छा, एक्चुअली यहाँ पे हमने प्लाज्मिड बनाया था और हमने एग्रोबैक्टीरियम में डाल के हमने सेल्स को ट्रांसफॉर्म किया, तो वो सेल ट्रांसफॉर्म हो गए। यहाँ पे हम एग्रोबैक्टीरियम यूज नहीं कर रहे। यहाँ पे आइडिया ये था कि एग्रोबैक्टीरियम में क्या करना है, प्लाज्मिड को डिलीवर करना है इनटू द सेल्स ऑफ प्लांट्स, इनटू द न्यूक्लियस ऑफ प्लांट्स। यहाँ पे हम प्रेशर के थ्रू, बम्बार्डमेंट के थ्रू हम इस डीएनए को डिलीवर करते हैं टू द न्यूक्लियस ऑफ प्लांट सेल्स। सो हम क्या करते हैं कि जी
तो सर, ये गोल्ड की ऐसी क्या प्रॉपर्टी है जिसकी वजह से हम इसे प्लाज्मिड के साथ कंबाइन कर रहे हैं?
हाँ, असल में अगर हम गोल्ड को या टंगस्टन को यूज ना करें तो हम इसको डिलीवर नहीं कर सकते, क्योंकि इसने सेल वॉल को भी ब्रेक करना है, सेल वॉल के थ्रू जाना है न्यूक्लियर मेम्ब्रेन के थ्रू न्यूक्लियस के अंदर। सो गोल्ड पार्टिकल और टंगस्टन पार्टिकल इज द मोस्ट सुटेबल वन, क्योंकि ये पार्टिकल साइज भी है ठीक, और ये जो है ये डिलीवर भी कर जाता है टू द न्यूक्लियस। इस पे एक्सपेरिमेंट्स हो चुके हैं दैट कैन डिलीवर दैट। ठीक है, ये इसकी एक वीडियो है, आई विल बी शोइंग दैट वीडियो, एंड ये चीज आपको क्लियर हो जाएगी कि क्यों ऐसा कह रहे हैं। ठीक है, तो third thing is electroporation. Electroporation is again the same thing, burst of electricity, और उसमें क्या होगा कि this is before the electrical field that has been induced. So this is the normal case before induction of electrical pulse. So these are genes, and we want these genes introduced into the cell उसमें, and this is electric E field, electric field. अब यहाँ पे क्या होगा जब इलेक्ट्रिक फील्ड हो जाएगी, सो दिस विल बी वन इलेक्ट्रोड एंड हियर विल बी अनदर इलेक्ट्रोड, और ये चार्जेस जो है ना वो सेपरेट हो जाएंगे, एंड दिस विल मेक फोर्स। जब फोर्स बन जाएंगे, सो आफ्टर पल्स, so the DNA will get into the nucleus, into the cell, और उसके बाद healing process again will occur, and now the cell are repaired and that can be regenerated. So this is the third method. So majority cases में Agrobacterium use होता है, या Microprojectile bombardment use होता है। Electroporation कम ही use होता है in plant case, use करते हैं लेकिन बहुत कम। So these three methods are used for plant transformation. एक और मेथड भी है polyethylene glycol, but उसको बहुत ज़्यादा ही वो conceptually है, किसी को यूज करते मैंने देखा नहीं है वैसे। So these three methods are used in plant transformation.
सर ये जो हम इलेक्ट्रोपोरेशन का मेथड यूज कर रहे हैं सर, ये हम कैलस पे अप्लाई करेंगे?
जी, ये कैलस पे भी अप्लाई कर सकते हैं, ये प्लांट टिश्यू पे भी कर सकते हैं, और इवन सस्पेंशन कल्चर सेल्स जो हैं उससे भी कर सकते हैं। ठीक है, शुड वी प्रोसीड?
जीन गन का एक मेथड हम लोग पूरा
ये जीन गन मेथड ही है। दिस इज एक्चुअली जीन गन, एक्चुअली दिस इज द जीन गन। जीन गन फिर डिफरेंट टाइप्स के आ रहे होते हैं ना, तो दिस इज द मोस्ट पॉपुलर जीन गन, और ये आपको मुख्तलिफ लैब्स में नजर आएंगी भी। आई हैव यूज्ड दिस थिंग, आई हैव यूज्ड दिस थिंग, the Agrobacterium and the gene gun method. So the microprojectile bombardment is through gene gun, this method. Okay. So अब अगर हम देख लें Agrobacterium को, Agrobacterium is a soil-borne pathogen, so it resides in the soil, it transmits through soil. So it is a plant pathogen, it infects plants, causes tumor, crown gall, so crown gall produce करेंगे, contain TI plasmid, tumor-inducing plasmid. This is TI means tumor-inducing plasmid, and they only infect dicots. Even though now people are are transforming monocots as well. So, but usually on this can only infect in naturally the dicot plants. So, dicots समझते हैं ना?
Yes, sir.
जिसमें दो cotyledons हो ना, and broadleaf plants हो. So how insects how they infect plants? So first is they need wound site, and through those wounds, Agrobacterium attached to the plant at the wound site, often at the base of the stem, and that is producing crown there, and the crown, and wounded plants secrete phenolic compounds, and these compounds activate virulence genes of Agrobacterium tumefaciens. So virulence genes will be there in the plasmid, and those virulence genes will be activated through phenolic compounds, and these phenolic compounds will be produced by the wounded plants, and wounded plants produce phenolic compounds. These phenolic compounds will activate the virulence genes within Agrobacterium, and that's then infect the plant. So this is how it happens. So this is the Agrobacterium tumefaciens cell, and this cell will be having the plasmid. Also, this is the bacterial chromosome, and this is actually the TI plasmid, and this is the transfer DNA. So only this region, the red region, will transfer into the plant cell. The remaining region will remain outside. So only this region will be integrated into the plant genome. So when this inoculated to a wound site, what will happen? This will producing crown gall, and the crown gall, when the cells were taken and when they were checked, the chromosome of the plant was having only this transfer DNA of this plasmid. This DNA was not there and this DNA was not there. Only this region, the red region, was integrated into the plant genome. So this is how it occurs and this is how it is. So there may be something here in the transfer DNA, how it integrates into the plant. So that is important. So that was studied, and after that, that was used for plant transformation. This was a best vehicle to transform our gene of interest. If we insert our gene of interest in this transfer DNA, it will transfer the same thing into the plant chromosome. So this was the basic concept that has been used later on to make transgenic plants, to make the genetically modified plants, and that's it. आज के लिए ये काफी है.
सर जो नेचुरल
प्लाज्मा
जी
इसका मैकेनिज्म पढ़ेंगे कि किस तरह से ये जो सेगमेंट है
जी ये सारे मैकेनिज्म्स हैं, आगे आ रही हैं, मैकेनिज्म्स भी आ रही हैं, सारी चीजें हैं. और हमने जी बायोलिस्टिक का भी पढ़ना है और इसका भी डिटेल से है सारा कुछ. लेकिन अभी मैं सोच रहा हूँ कि टाइम हमारे पास
सही है. प्लाज्मिड है, ट्यूमर प्लाज्मिड, तो सर जो इंटीग्रेशन साइट्स हैं जिसके जरिए ये प्लांट क्रोमोसोम में इंटीग्रेट होता है, तो सर वो भी हमें जीन ऑफ इंटरेस्ट के साथ इंजीनियर करनी पड़ती है वो, या वो हम इसकी इंडिजीनियस यूज करते हैं जो नेचुरल है?
वो इसके नेचुरल यूज कर रहे हैं. वो इस असल में हम कर क्या रहे हैं, यहाँ पे एक जीन इंटरेस्ट इंसर्ट कर देते हैं. इसके जो राइट बॉर्डर और लेफ्ट बॉर्डर है ना, ये दोनों बॉर्डर्स जो हैं, लेफ्ट बॉर्डर राइट बॉर्डर, वो इंपॉर्टेंट है. उसके सीक्वेंसेस हम आगे पढ़ेंगे, वो इसके सीक्वेंसेस हैं प्रॉपर के, वो सीक्वेंसेस जो है ना वो दे आर इम्पोर्टेंट. और उस सीक्वेंसेस को सिर्फ राइट बॉर्डर और लेफ्ट बॉर्डर को छोड़ के बाकी हम सारा ये चेंज करेंगे, क्योंकि ये क्राउन गॉल किस तरह बना रहा है, ये ट्यूमर किस तरह इंड्यूस हो रहा है, वो भी हम पढ़ेंगे इसमें डिटेल से. तो होल्ड ऑन एंड वी विल बी स्टडीइंग दैट इन डिटेल एक्चुअली. ठीक है. Up till now, it's clear.
Yes, sir.
ठीक है. So अभी तो हमने 15 slides पढ़ी हुई हैं. We have many more. So on the same topic, हमारे पास बहुत सारा है इस पे काम. So we will be studying this. हो सकता है next class में भी ये खत्म ना हो, उससे के बाद भी चलें, क्योंकि I want कि हम chloroplast transformation भी पढ़ लें. So chloroplast transformation भी है, ये हम डिटेल से कवर करेंगे. फिर हो सकता है हम chloroplast transformation भी कवर कर लें. And that is important because this will having more issues. Chloroplast transformation में maternal inheritance है, जिस तरह mitochondrial है ना. So chloroplast में maternal inheritance है, and that is more beneficial, and that is more having more edges over the the nuclear transformation. So this is nuclear transformation ना. ठीक है.
जी सर.
So for now, आज डेट क्या है? 22.
Yes, sir.
Okay, Aisha Lodhi.
Yes, sir. Present.
Present है. Ali Raza. Ali Raza नहीं है. Okay. Absent. So Sarish Said.
Yes, sir.
Kokab Imran. Hassan Raza.
Yes, sir.
Huma Majid.
Present, sir.
सोलत नवाज, सोलत नहीं है,
प्रेजेंट है।
प्रेजेंट है। ओके, दुर्र रमीशा,
प्रेजेंट।
ओके, सिमरा आरिफ,
प्रेजेंट सर।
मरियम,
यस सर, वो थी, उसका डिस्कनेक्ट हो गया है।
ठीक है, जवेरी अरशद,
यस सर, प्रेजेंट।
महनूर,
प्रेजेंट सर।
खदीजतुल कुबरा,
यस सर।
लाइबा ताहिर, लाइबा ताहिर नहीं है,
यस सर।
है लाइबा,
यस सर, उसकी नेट प्रॉब्लम हो गई है कोई, इस वजह से यार।
सवेरा असगर,
यस सर।
वजीहा उरूज, वजीहा नहीं है, एंड ताबिश आरिफ, ताबिश आरिफ,
यस सर।
ताबिश आरिफ है,
यस सर, ताबिश प्रेजेंट।
एंड समैया, समैया सजाद,
सर। प्रेजेंट।
ओके, सितरा नूर,
यस सर।
मारू खालिद,
प्रेजेंट सर।
निमरा,
प्रेजेंट सर।
नुमान अजीज, मोहम्मद नुमान अजीज आया हुआ है, आज है, एब्सेंट है, नुमान आया हुआ है?
सर वो स्टार्ट में आया था।
ओके ओके, अदीन खान, अदीन is absent, अफीफा जुल्फिकार, अफीफा जुल्फिकार प्रेजेंट है, आफाक अहमद,
यस सर, प्रेजेंट।
ओके, शहजादी सखी,
जी सर, प्रेजेंट।
he's present today, okay, महनूर खान,
प्रेजेंट सर।
and आयशा जमान,
यस सर, प्रेजेंट।
ज़ैनब हयात,
प्रेजेंट।
रोहा अहमद,
प्रेजेंट सर।
मारिया उस्मानी,
प्रेजेंट।
जवेरिया इफ्तिखार,
सर वो प्रेजेंट थी, डिस्कनेक्ट हो गया उसका।
अरीक मतीन,
सर प्रेजेंट थी वो, अभी तो देखा मैंने।
फैसल बशीर,
सर वजीहा प्रेजेंट।
वजीहा कहाँ पे गुजरा, वजीहा उरूज,
सर यू
ठीक है, ओके,
सर सोलत भी प्रेजेंट है।
सोलत प्रेजेंट है,
जी सर।
कहाँ पे सोलत, हाँ प्रेजेंट लगाया हुआ है, वो मुझे किसी ने कहा था सोलत, ओके, सो फैसल बशीर, फैसल बशीर नहीं है,
फैसल आया हुआ है।
आया हुआ है, ओके, अफजाल यूनस,
यस सर।
एंड रेशमा,
सर रेशमा का डिस्कनेक्ट हुआ है।
ओके, अब्दुल राफे प्रेजेंट, नईम, नईम, नईम नहीं है, ओके, एब्सेंट,
सर उसके इंटरनेट का इशू है।
आया हुआ था, नईम आया था,
शुरू में कनेक्ट हुआ था लेकिन वो ना फिर डिस्कनेक्ट हो गया शुरू में।
कामरान भट्ट, ठीक है,
कामरान भी आया हुआ है।
अमतुल रहमान,
प्रेजेंट सर।
ओके, सज्जल वकास,
प्रेजेंट सर।
जैन अब्बास, जैन इस
Yes,
प्रेजेंट,
sir.
वो सबसे पहले आया था जैन, और उस वक्त जैन और कोकब आए थे ना,
Yes, sir.
so कोकब is the person who signed in 10 minutes before you and usually in the class. Okay,
सर मार्क्स असाइनमेंट्स की जो क्विज़ लास्ट टाइम लिए सर उनको आप अगर एलएमएस पे अपलोड कर दें।
LMS असल में यहाँ पे मेरे पास मैंने LMS पे किए हुए हैं वैसे।
सर अपलोड हुए हैं?
हाँ हाँ मैंने कर दिए हुए हैं।
सर चेक कर लो।
हाँ ठीक है। वो असल में पोर्टल मुझे एक्सेस नहीं है क्योंकि यूनिवर्सिटी से बाहर पोर्टल एक्सेस नहीं है। सो LMS है, LMS मैंने कर दिया। मैंने कहा चले शो हो। मार्क्स जो है ना वो टीए ने चेक किए हैं। सो आई डोंट नो कि क्या क्या मार्क्स आए हुए हैं। ठीक है आप देख लें। इट हैज बीन अपलोडेड। सो आज एब्सेंट जो है वो अली रजा है और अदीन खान। So, okay, thank you so much, everybody. Okay, Allah Hafiz, Allah Hafiz.
Allah Hafiz.